My name is Leslie Bernstein, and as co-chairperson of the AACR Cancer and Epidemiology Award Selection Committee, I have the honor of opening this session and citing the outstanding accomplishments of the winner of the 9th AACR-Prevent Cancer Foundation Award for Excellence in Cancer Prevention Research. Before I introduce the recipient of this honor, I would like to invite to the podium Ms. Christina Hunken, who is the Manager of Research Administration and Community Outreach for the Prevent Cancer Foundation in Alexandria, Virginia. The Prevent Cancer Foundation is the co-sponsor of this prestigious award.
Good evening. In 1984, Carolyn Beau Aldeget founded our organization in memory of her father who died of cancer. 2 years later, the Prevent Cancer Foundation made its very first research grant to NCI fellow Dr. John Sandlin in the amount of $8,000 to support his research on Burkitt's lymphoma. This grant helped provide a 4th year of funding to sustain Dr. Sandlin's work. The Prevent Cancer Foundation is now celebrating its 25th anniversary, and we are thrilled to report that the foundation has provided more than $120 million in support of cancer prevention and early detection research, education, and community outreach programs across the country. On behalf of Beau and the foundation, we congratulate Dr. Groopman on his vast accomplishments, and we are honored to be a part of his future achievements in cancer prevention. Thank you, Dr. Groopman. And thanks to all of you for the work that you do every day to make this world free from cancer. Thank you.
Thank you, Ms. Hunken. And on behalf of the American Association for Cancer Research, I would like to thank the Prevent Cancer Foundation for its continued support of this distinguished award. The AACR-Prevent Cancer Foundation Award for Excellence in Cancer Prevention Research recognizes a scientist residing anywhere in the world who has made seminal contributions in basic, translational, clinical, epidemiological, or behavioral research in the critical field of cancer prevention. These studies must have had a major impact on the field and have stimulated new directions in this important area. The Award Selection Committee, which reviewed many nominations for this award, has chosen to recognize Dr. John D. Groopman, Ph.D., Associate Director of Cancer Prevention and Control at the Johns Hopkins Kimmel Cancer Center, and the Anna M. BEIJING, I should have asked you how to pronounce that word, John, Professor and Chair of the Department of Environmental Health Sciences at the Johns Hopkins Bloomberg School of Public Health in Baltimore, Maryland. Dr. Groopman is recognized for his outstanding work in the discovery, validation, and application of molecular biomarkers to probe the etiology of liver cancer and the means to prevent it in high-risk populations in the developing world. The primary focus of Dr. Groopman's research is hepatocellular carcinoma and the rapid translation of the initial biomarkers he discovered, which were rapidly translated into a multinational investigation of the etiology of hepatocellular carcinoma that, for the first time, characterized the relationship between exposure to the mold-derived food contaminant aflatoxin and infection with hepatitis B virus. I should tell you that I learned all about what he was doing through my close colleague, Ron Ross, with whom he worked. Dr. Groopman's seminal work on the etiology of hepatocellular carcinoma through his biomarker approaches in his translational research efforts on chemoprevention are bringing new prevention opportunities to high-risk populations in the world. Today, we are privileged to recognize Dr. Groopman for his exemplary contributions to cancer prevention and his dedication to this field by naming him the recipient of the 2010 AACR-Prevent Cancer Foundation Award for Excellence in Cancer Prevention Research. Congratulations, Dr. Groopman.
We didn't plan this.
No, not well enough.
Thank you very much, Leslie, for the kind introduction. And in particular, I think it's very important to recognize that all the work that I'm going to share with you over the next 40 minutes or so is really the outcome of a tremendous amount of teamwork and collaborations that have developed between epidemiologists, toxicologists, chemists, and clinicians to deal with the issues of not only hepatocellular carcinoma, but the application of these types of biomarker strategies to high-risk populations around the world. And again, I do want to emphasize that it is the team and not I. So if I can have the first slide. Okay. First and foremost, the Prevent Cancer Foundation has been a long-standing supporter of cancer prevention research, the partnership with AACR, and just the outpouring of philanthropic donations and talents that are brought to bear to raise the recognition of the importance of cancer prevention is something that I deeply appreciate, and I think all of us at this meeting deeply appreciate all the efforts on the part of the Cancer Prevention, Prevent Cancer Foundation. At the same time, I also want to acknowledge that through my entire professional career, AACR has been the steadfast organization that has really contributed to my own academic and career development. From the very first meeting that I attended in 1975 to the present, AACR has really been an important part of my career. I have absolutely no financial relationships to disclose. I will not discuss any off-labeled or investigational use of various drugs. Now, what I do want to do is I want to share a story with you. I want to share sort of a journey, if you will, that really starts with the ABCs. And the A part are agents, B are biomarkers, and C are cohorts. And what I'd like to do over the next few minutes is intertwine these ABCs in terms of how we developed a strategy to validate a variety of biomarkers that I'll be sharing with you. All of this starts with epidemiology. All of this really starts with trying to understand the exposure relationships, how they interact with various genes, and what the disease linkage is. We are driven in this particular type of strategy to investigate people at the highest risk for cancer so we can begin to translate our findings into prevention studies. Again, this now involves the identification of biomarkers and the development of quantitative analytical methods for their detection. These biomarkers need to be validated and investigated in both experimental animal models along with human investigations, and there needs to be an interaction of the feeding of knowledge that we gain from both these types of investigations in order to forward the field. I'll share with you some of the strategies that we've employed to determine the relationship of biomarkers to disease outcome, and at the same time to ask questions about what happens to these biomarker levels when we modulate risk in experimental models. In other words, if we lower the risk in experimental models, do the biomarkers track with outcome? At the same time, funneling down into this area of a validated biomarker is really the need to have the cross-sectional and longitudinal studies in high-risk human populations, and then the further investigations in case control, cohort, and clinical trials. And I'll touch upon each of these points as I evolve this story. Our interest is in hepatocellular carcinoma, and as you can see from this slide, hepatocellular carcinoma is very much overrepresented in sub-Saharan Africa and here in East Asia and Southeast Asia. Indeed, hepatocellular carcinoma is a devastating disease in these very economically poor regions of the world. And just to give you a a sense of the global impact of hepatocellular carcinoma, there are at least 695,000 deaths each year. This is clearly an underestimate due to the inability to completely ascertain all the cases that occur worldwide. And just as a proportion, you can see that the population of Baltimore City in 2008 was about 637,000 people. So again, conceive of a city of Baltimore population equivalent dying of liver cancer each and every year. And I think that will start to reflect some of the scale of the impact of this disease. At the same time, what is of intense importance to us in the public health community is that this is a disease where the median age of diagnosis is between 45 and 50 years of age. And the median time, as you'll see, from diagnosis to death is measured on the order of 3 to 6 months. So this is a disease of early life as we view it. It is a disease that, again, has a devastating impact in many regions of the world. So let's first look at agents. When one looks at the hepatocellular carcinoma incidence data that I just shared with you, you can see to the first approximation there is a bit of a concordance with the prevalence of the hepatitis B virus in many areas of the world. And these types of data provided some early insight into the relationship between the hepatitis B virus and the occurrence of hepatocellular carcinoma. Again, in perspective, HCC is the third leading cause of cancer death worldwide. Over 80% occurs in the economically developing world. And since there are greater than 400 million hepatitis B virus carriers in the world, this will probably translate into upwards of 100 million liver cancer deaths over the next quarter century. So again, this is a disease of tremendous public health impact that is superimposed upon a developing part of the world who is now on the verge of economic growth over the next century. The other part of the story is aflatoxin. Aflatoxin was originally discovered in the United Kingdom in 1960 from a moldy toxic animal feed. This is actually a picture of the original sample of the Rossetti meal from which aflatoxin, the toxic principle, was eventually elucidated. Aflatoxin is found as a frequent contaminant of improperly stored foods around the world. These spores are universally distributed in the soil. The mold can readily grow both during and after harvest of the grains. Some of the relevant properties of aflatoxin: it's highly fluorescent, and this led to some of the original, if you will, biomarker detection strategies for the measurement of aflatoxins in food. It is exquisitely lethal to a wide variety of animal species. It's also pretty much an equal opportunity carcinogen in that virtually every animal species for which it's been tested, we've found that it causes cancer primarily to the liver. It is also immunotoxic to both animals and humans, and indeed there are a number of studies from some of our colleagues, Chris Wild, who's now at IARC in particular, that has related aflatoxin exposure to stunting and growth defects in children who are exposed to high levels of this toxin in West Africa. So it has tremendously wide-ranging toxicologic along with carcinogenic properties. Aflatoxin, when it was initially isolated and discovered, clearly caused liver cancer in experimental animals, but the pathway through which it would traverse, starting off as a parent compound and leading to the formation of a tumor, had really not been understood and required several decades of investigation. All of this really starts with a partnership in chemistry and toxicology that was pioneered by the late George Bushy in the Department of Chemistry at MIT and continued with Jerry Wogan's work also at MIT in the toxicology. Here I'm showing actually the original structures that George wrote, as you can see, on March 22nd, 1963, when he was able to structurally elucidate the structure of aflatoxin B1 using NMR strategies that was state-of-the-art in the early 1960s. But it was from this chemistry and from then exploring the toxicological properties of these pure compounds that really all of the work that I'm going to describe on the aflatoxin side of the paradigm developed. And that leads to the second part, the biomarkers for investigations in these high-risk populations. The landmark studies from Taiwan, from Palmer Beasley and his colleagues, revealed in the early 1980s this absolutely stunning relationship between those individuals who are hepatitis B surface antigen positive, who then went on to develop hepatocellular carcinoma. So this was a large prospective study of nearly 23,000 men in Taiwan, and you can see that the relative risk for the development of HCC was almost 100 in this population. And indeed, this really represented the power of an immunological biomarker to begin to provide insight into the etiology of hepatocellular carcinoma. When one started to tease out some other data from different populations around the world, it became quite clear that while the hepatitis B virus was incredibly important, there was wide variation in HCC rates that could not be explained by this one metric alone. And this, in turn, is what has led to a variety of investigations to tease out what, in combination or independent thereof, of the hepatitis B virus was contributing to these high rates of hepatocellular carcinoma. And that's really where the biomarker strategies for the aflatoxin adducts began to play a role. And so here we now know that the parent compound needs to be activated through some cytochrome P450 mechanisms to an exoepoxide in particular that forms a pro-mutagenic DNA lesion. This is the structure of the aflatoxin DNA adduct. Indeed, virtually all the DNA damage mediated by aflatoxin occurs through the formation of this Aflatoxin-N²-Guanine adduct. The chemistry of this interaction does in fact lead to a variety of other secondary products that have other biological, both toxic and mutagenic properties. But again, it is this exquisite formation of this N²-Guanine adduct through a three-dimensional structure of how a unique metabolite of aflatoxin, this exoepoxide, is able to slide into DNA and nuzzle up very closely to the N7 position of guanine, forming this covalent DNA adduct. The chemistry of this has proven to be very, very important, and indeed understanding the three-dimensional structure now provides us insight as to why aflatoxin binds to DNA at least an order of magnitude better than any other bulky carcinogen. Again, the resultant biology from the knowledge of this DNA adduct needed to be elucidated, and I'll take you through some of that story. In our lab, we pride ourselves on the development of quantitative measurements, and this is some recent data from our lab that looks at the ability to measure the Aflatoxin-N²-Guanine adduct that I just described, along with one of its major derivatives, the formin-aminopyrimidine adduct, in both human and experimental rat urine. All of this work is really led by Patricia Egner and Marlon Friesen in the lab, and through their partnership in combining efforts in these types of analytical measurements, we now have a rather important outcome, which I think is important for this group, in that our non-detects for the measurement of the Aflatoxin-N²-Guanine is now less than 5% of the people that we monitor in these high-risk regions of the world. 15 years ago, our non-detect rate was about 40%. Now, when you have a non-detect rate of 40%, that gives an enormous amount of latitude with respect to having misclassification of exposure and clearly misclassification of risk. So our ability to have increased analytic sensitivity, the ability to quantify these types of adducts in these biological samples has now resulted in having essentially a complete capability of monitoring this important DNA lesion in both animals and humans. The acquisition of these analytical technologies now makes it readily available for us to measure this adduct in urine, hence the urine green color here. We can measure other metabolites such as the mercapturic acid in urine that reflects glutathione transferase mediated reactions, along with an aflatoxin albumin adduct that is readily available in plasma. But again, we want to know more about the resultant biology that occurs following this DNA damage. And this has again been revealed through a number of our experimental and human investigations. And that comes to the third part of the ABCs, which is the cohorts. And if I'm permitted a little bit of latitude, I want to talk about animal models as a cohort along with human investigations. So here on the animal model side of things is really asking two very simple questions. The first is, what is the relation of a given biomarker to disease outcome, something that we can readily monitor and measure in experimental models? And then two, if we modulate the risk of the animal, in other words, if we lower their risk of developing disease, does the biomarker track with that particular outcome? If the biomarker stays the same, then it's really not going to have very much utility in understanding risk. If it doesn't track with outcome, then we can at least have some insight into the predictive value of this particular biomarker. This work has been a partnership, and this is a partnership of 3 graduate students from Jerry Wogan's lab, Tom Kensler, who's sitting here in the audience, and Bill Roebuck, who's at Dartmouth Medical School. And for the students in the audience, I'd just like to remind you and encourage you that some of your lifetime colleagues in science are going to emerge from your graduate years and from your postdoc years. So try not to make enemies out of them. These are individuals who can really be your steadfast colleagues through decades of scientific career. I had a little bit more hair then. So the need to develop a quantitative rat model to test these questions of biomarkers was really something that we started to discuss at an AACR meeting in San Diego in 1983. We actually did talk science during the meeting. And one of the questions was, if we know that the aflatoxin DNA damage product occurs within hours of exposure, and we also knew that there were preneoplastic lesions that were predictive of cancer risk that emerged after weeks following exposure, and if we had to wait the months or indeed year and a half prior to liver cancer, could we in fact develop strategies that could test biomarkers and look at how they would track and predict outcome? Initial studies were to ask a very simple question, and that was, in the face of a chemoprotective agent, this is dithiothione, which is the parent molecule of ultipraz, that if you gave multiple doses of aflatoxin and asked the question, in the face of the chemoprotective agent, did you in fact see reduction in DNA adduct levels? And indeed we did. We were able to reduce DNA damage in these animals between 80 and 95%. And so to the first approximation, our understanding that the aflatoxin DNA adducts might have predictive value was certainly starting to be validated. At the same time, we found that the GGT foci in these experimental models could be dramatically reduced by the chemoprotective intervention. And as you can see here, ultipraz virtually wipes out these preneoplastic nodules. At the same time, carrying out a lifetime study in animals, we found that ultipraz was 100% effective for chemoprotection against aflatoxin-induced hepatocellular carcinoma in rats. So by tying together these models of the preneoplastic lesions, liver cancer outcome, and DNA adductology, we were able to begin to structure our strategies of using these biomarkers not only to test the efficacy of these types of intervention strategies in experimental models, but to begin to propose the application of these to high-risk human populations in clinical trials that Tom Kensler has been leading. One of the findings of the studies that emerge out of these experimental results was something that we have never forgotten, and that is that in the face of getting over a 95% reduction in GGT foci in this experimental model, if we then ask what was the inhibition of the aflatoxin DNA adduct damage, you can see that the reduction of about 70% of adduct damage conferred a greater than 95% protection in these animals. And indeed, as you look at these data, one of the things that emerges is that very attainable reductions in DNA damage can have a much greater impact on the protection of those animals from carcinogen exposure. And indeed, the aflatoxin DNA adduct levels underestimate the anti-tumorigenic efficacy of ultipraz. So we think that this is important because, again, the ability of reducing adduct burden by 50% may confer an extremely high protection, not only in these experimental models, but something that we can test in human populations. The human studies required, again, large-scale collaborations. Leslie talked about the studies that we had with Ron Ross and Brian Henderson at USC, along with Mimi Yu, and a number of our collaborators at the Shanghai Cancer Institute, in particular Yutang Gao. And all of these investigations tied together how we could apply our biomarker strategies in cross-sectional and longitudinal studies in people, along with case control, cohort, and clinical trial investigations. So when we look at the human studies in terms of cohorts, there are a number of cohorts that we've been involved with over the years, and I'm just going to use some small vignettes from these investigations to give you a sense of how we've applied these types of technologies. All of this work really starts around 1980 in China, where if you look at the liver cancer map generated in the mid-1970s in China, there's a strikingly high level of hepatocellular carcinoma in these coastal counties in China and here in the Guangxi Autonomous Region. So some of our original work was in Guangxi, where townships suffered more than 15% of the people dying of hepatocellular carcinoma. Again, over half the liver cancer cases in the world are diagnosed in China. Half the deaths also occur in China. So it's the second leading cause of cancer death at this point in time. Some of our early studies in Guangxi revealed that we were able to measure the Aflatoxin-N²-Guanine in people. This was a bit of a breakthrough in the fact that we had data to show that we could measure this in experimental rat urine, but this was the first study that really allowed us to know that we could in fact measure this DNA damage product in human urine, and indeed there was a strong relationship between increasing exposure in the diet and increasing levels of the Aflatoxin-N²-Guanine adduct in urine. We did the dietary work by doing split bowl analysis, weighing the food that people were consuming, and then going back to the lab and analyzing for the exposure to aflatoxin B1. We were privileged to be involved in the Shanghai Cohort. This is 18,244 men who were enrolled starting in 1984 into 1985. And some of the original work, and I remember these meetings very distinctly, when I thought that people wanted me to measure 18,244 urines. And I thought, well, that's going to take a while. And so it was really beginning to understand that using a nested case control study design, allowing for some biostatistical strategies to work to our benefit in order to design precise studies to test the relationship between the hepatitis B virus and aflatoxin in the development of hepatocellular carcinoma. And here in the work first published and led by Ron Ross was this very powerful greater than multiplicative interaction between the hepatitis B virus as measured by the by the hepatitis B surface antigen and aflatoxin biomarkers in the urine, and so the relative risk turned out to be 59. This is statistically significant. For aflatoxin alone, the relative risk was 3.4, also statistically significant, and for the hepatitis B surface antigen, it was also statistically significant at 7.3. These data have also been replicated in Taiwan, showing this very powerful interaction between aflatoxin and the hepatitis B virus in the development of HCC. But putting my public health hat on for a moment, I would also like to submit to you that this means that we have insights in terms of developing strategies for prevention that can employ things such as the vaccine against hepatitis B virus, but other strategies that could blunt the impact of aflatoxin that also should have a powerful positive impact in reducing hepatocellular carcinoma in these high-risk populations. So when we go to some of the cohorts that we've been involved with in Qidong, I'll share some of the data from those investigations with you. And one of the striking features about Qidong, it's here, this county in the Peninsula region right across from Shanghai. This is Chongming Island, the Yangtze River. The geographic pathology of hepatocellular carcinoma in Jiangsu is quite striking. You can see that within a 200-kilometer range of Qidong, there's a greater than 25-fold range of hepatocellular carcinoma diagnosed. And indeed, the hepatitis B surface antigen positivity rate is identical across Jiangsu province. So the median age of liver cancer death is between 45 and 50, and again, survival is really quite poor. As we carry through the paradigm of what are the biological consequences of aflatoxin DNA damage, it was really the insights from a large international collaborative investigation that continues to this day, beginning to report on p53 mutations in hepatocellular carcinoma. These pie charts just illustrate the different mutations that are found in these HCC, and it's really the pioneering work of Kurt Harris, who really led some of the initial investigations that characterized a codon 249 mutation in p53 from liver tumors here in Qidong. I also acknowledge Kurt not only for the work that he did on p53, but he was one of my mentors when I was a staff fellow at the NCI in his lab a little while ago. So this is a landmark in the sense that we have two findings from this p53 work. One is that a number of strong mechanistic studies have now linked aflatoxin DNA damage to the formation of this codon 249 mutation in p53. And from a biomarker perspective, we and others are now using these codon 249 mutations as biomarkers to detect the biological consequences of DNA damage mediated by aflatoxin in human plasma samples. Indeed, Marlon Friesen in our lab, working in collaboration with Bert Vogelstein's lab, have developed a quantitative methodology, which we call SOMA, for short oligonucleotide mass analysis, to measure codon 249 mutations in biological samples. The plasma is taken, DNA is isolated. Again, many of you are well aware that these fragments of DNA end up in circulation, so we can trap them and measure essentially things that occur in target organs by looking at the shed DNA out in circulation. And then using a number of molecular biological and quantitative mass spectrometry techniques, we're able to measure the levels of the 249 mutation in these biological samples. For the analytical chemists in the audience, you recognize the need to apply an internal and standard into these samples, allow us to look for recovery and give very precise data with respect to the levels of these mutations that we're able to measure. And this is an example of measuring 40 copies of the 249 mutation by the SOMA technology. So again, I want to stress the need for quantitation, because this is the way we can start to compare and track people from time to time and compare across individuals. So from this particular set of strategies, we now know that the p53 mutations in codon 249 play a very important role in hepatocellular carcinomas in Asia and in sub-Saharan Africa. The ability of having a biomarker reflective of this biology is something that we and others have attained. The biological knowledge about how p53 works is being continually elucidated, and this particular pathway of the aflatoxin HCC outcome is something that we have come an incredibly long way over the last 20 years, and we're really beginning to put some final punctuation marks on this particular part of the paradigm. But at the same time, there's the hepatitis B virus. So starting about 10 years ago, recognizing that we couldn't just do aflatoxin alone, we needed to understand more about the the interaction between aflatoxin and HBV in the development of hepatocellular carcinoma, we started a number of investigations to see if we could tease out some of that biology. The hepatitis B virus work is again driven by our samples that we have from our cohort investigations, primarily in Qidong. This is a cartoon of the structure of the hepatitis B virus, and I'll just note that what we have been very much focused on are a series of mutations that are acquired by the HBV virus. In particular, this particular mutation that I'll share with you, the 1762-1764 double mutation right here in the X gene part of the hepatitis B viral genome. Indeed, the biology of hepatitis B virus X gene and its protein is really quite amazing. This particular protein, as shown in this diagram, is involved in many, many different impacts on the biology of cells, from DNA repair to cell cycle effects to inducing apoptosis. There's a lot of biology that is now being teased out about the X gene of hepatitis B virus. At the same time, the formation of this mutation, this double mutation in the X gene, confers the production of a mutant protein, and that mutant protein has very different biologies that we also feel is very important in the embellishment and the development of hepatocellular carcinoma. So we're intensively on the trail of trying to understand more about the biology of this particular mutation, how it's acquired, and see whether we can in fact reverse it. Our initial investigations involved an examination of 70 hepatocellular carcinomas from Qidong, and we found that 3/4 of those tumors in the tumor DNA contained integrated HBV virus, where that integrated virus had this double mutation in the X gene. So when you see 3/4 of the samples being positive, this immediately starts to tell you that this could be an important signature of exposure and potentially risk. In collaboration with Jinmin Wang now at the University of Minnesota and going back to the Shanghai Cohort, we asked a very simple question, and that simple question was, among individuals who are hepatitis B surface antigen positive at baseline, who then went on to develop hepatocellular carcinoma, in those individuals, was there any increased risk, if you will, conferred if you were positive in the plasma sample for this 1762-1764 DNA fragment? And as you can see, there was about a 3-fold increased risk for the development of hepatocellular carcinoma in those individuals who are already at risk because they were hepatitis B surface antigen positive. So this is starting to hone in on those individuals who may be, at, if you will, at the highest risk for the development of disease. We're privileged to, again, continue our collaboration with Chen Guo in Qidong, and Chen Guo did a remarkable thing as part of a screening effort in 1989, where 36,000 men in Qidong were screened for the hepatitis B virus. This was part of a study to identify those individuals who could then be vaccinated using the HBV vaccine. What Chen Guo did was identify the 5,581 men who were surface antigen positive in 1989. These people were enrolled into a cohort and have been followed ever since. Strikingly, as you can see, 773 of these men, as of the end of last year, had in fact died of hepatocellular carcinoma. So that's 773 out of a little under 5,600. Again, I think it illustrates the enormous impact of this disease in this population. In our initial study, we were able to recover from those 1989 samples, in about 69% of the samples, we were able to recover DNA from these original serum samples. And as you can see, 83% of the DNA samples that we were able to recover here contain the detectable levels of the 1762-1764 double mutation. Again, this is illustrating that not only are these individuals hepatitis B surface antigen positive, but at some point in time, they acquire these mutations. We think that this causes a change in the biological consequences of the HBeAg protein, but at the same time, there is this extraordinary overrepresentation in these individuals who went on to develop HCC and died from the disease in this particular cohort. Alvaro Munoz in the Department of Epidemiology at Hopkins refined a strategy to do a match case control study utilizing the samples. These are data that we just decoded about 2 weeks ago, so I'm able to share with you hot off the press data. We found that about 345 of these cases could be PCR amplified. And again, 81% of these cases were positive for the HBV double mutation in codon in nucleotide 1762, 1764. The match controls drawn from this population, again, matched on age, time of screening, and other parameters. We found that 40% of the match controls were positive at baseline for this particular double mutation. And the match odds ratios for the development of HCC was 6.7. So again, here we have individuals who in 1989 were screened, blood samples were ascertained, and now we're monitoring the occurrence of this particular mutation up to 20 years prior to liver cancer diagnosis, and seeing that there was an extremely high increased risk for the development of hepatocellular carcinoma in these individuals who had the misfortune of developing this double mutation. We further found, again, in keeping in mind that this is a match case control study, we then subsequently found that 20 of these 250 individuals subsequently went on to be diagnosed and die from hepatocellular carcinoma. So I'll share with you some of the quantitative data that we've just decoded from this particular investigation. This is a modified box plot. So here is the 50% median, 75%, 90%, and so forth. For those of you, just to orient you to the y-axis, this is PCR cycles. So for every increase of going from, let's say, 30 to 31, that's a twofold difference. 30 to 32 would be a fourfold difference, and so forth. We don't normally think in this doubling time, but that's essentially what's occurring on this particular scale. And you can see that in the HCC cases in this cohort, the median for these particular P53 HBV mutations became positive after 30 cycles. In the controls, it was about 33.8 cycles. And so in the cases, there was a 13-fold higher level of these particular mutations in plasma at baseline of those individuals who eventually went on to develop hepatocellular carcinoma. Again, this is beginning to tease us into thinking that there is a quantitation involved, that there's a tracking sort of mechanism that we can use in looking at the levels of this mutation to predict risk and eventually development and outcome in those individuals who are at highest risk of developing HCC. Very strikingly, we then explored those 20 individuals who emerged out of this match control and asked, was there anything unusual about those individuals who then subsequently, over the following few years, went on to develop hepatocellular carcinoma? And the thing that was really striking was that those individuals, their median was 275-fold higher in the level of the 1762, 1764 double mutation in their plasma. So again, collectively, we think that these types of studies and beginning to put some quantitative analysis around these types of biomarkers could begin to start to tease out who is at highest risk for the development of disease. So again, the paradigm of the aflatoxin HBV story is a paradigm of probably parallel biologies. we have a very powerful, very strong DNA damaging agent that can target a very important gene in the development of human hepatocellular carcinoma that in turn confers risk for the development of this disease. At the same time, we have individuals who are exposed very early in life to the hepatitis B virus. The acquisition of these mutations changes and embellishes the biology of this virus. All of this teases into chronic inflammation, cell turnover, a number of the proliferative cycles that undoubtedly feed back and further the development of this disease. And for those individuals who have both of these types of exposures, they're going to be at exquisitely high risk for the eventual development of hepatocellular carcinoma. These biomarkers of aflatoxin are short-lived biomarkers that track with the exposure, and indeed in many of the populations that we work with, because economic change has not occurred at as a rapid rate as one might have hoped, these types of biomarkers, because there's year to year the same level of high exposure to aflatoxin in the diet, in fact do give us a handle on both current and past exposures to this particular carcinogen. The biomarkers of P53 are longer lived. Indeed, we and others can detect this particular mutation in plasma at least 5 years prior to hepatocellular carcinoma diagnosis. And again, this helps to integrate aflatoxin exposure and fix an important biological event in the development of disease. We're exquisitely interested in these mutations in the hepatitis B virus. The fact that we can detect this mutation in plasma up to 20 years prior to HCC risk tends to suggest to us this is a very early event in the HCC process, and indeed is something that confers almost catastrophic risk for those individuals who begin to develop this particular mutation. It is still unclear how this particular double mutation arises, and this is an area of active research in our lab and others. So the final component is really prevention, because to do all of this analytical work, to do these collaborative studies, to do all these investigations without having the opportunity to try and translate this to those individuals who are at greatest risk of the disease would really be an unfulfilled and unfulfilling part of the story. When one looks at Qidong and BEIJING, there's this striking finding that in Qidong, liver cancer is diagnosed on average 25 years in people who are 25 years younger than in BEIJING. Under the microscope, these are the same tumors. They're hepatocellular carcinoma. There's very little obvious differences between these types of tumors, but the striking feature of having a much earlier onset and diagnosis of disease clearly points to environmental factors playing an important role. We know that early life exposures are incredibly important in the development of this disease. HBV infection often occurs before age 1 in both these Asian populations along with populations in sub-Saharan Africa. It really compels the need to truly have the global vaccine trials and implementation of vaccine studies against HBV. But unfortunately, this is going to have to continue essentially at the birth rate of these high-risk populations, and that really means for the rest of the 21st century there are going to be people who are going to be at risk from HBV infection. At the same time, aflatoxin exposure is also something that occurs not only starting in early life but occurs in utero. Chris Wild, one of our colleagues, has demonstrated that the fetus has metabolic capability for activating aflatoxin B1 to damaging species, and certainly as children begin to eat the adult diet, which they do very early in life in these populations, they're going to be exposed to this ever increasing and decreasing cascade of aflatoxin exposure. So there are strategies for the prevention of hepatocellular carcinoma. No one alone is going to be enough. This is going to have to be truly a use of a number of different strategies. Clearly, the immunization with HBV vaccine is one important starting point, but it's not the end. We still have those 400 million carriers in the world, and we still have populations yet to be born that will not be able to avail themselves of the vaccine because of the types of financial support and public health infrastructure necessary to get those vaccines to the populations at risk. I would submit to you the primary prevention in reducing aflatoxin consumption, again something that our colleague Chris Wild has pioneered, is also an important adjuvant in these populations where a single staple grain is a main source of all their dietary exposure. Secondary prevention using chemopreventive strategies is an important adjunct to our armamentarium in dealing with these high-risk populations. Again, Tom Kensler has led our efforts in the UltraPraz clinical trials, recent trials using broccoli sprouts as a form of introducing sulforaphane into the diet, chlorophyllin, and there are several trials in various high-risk populations that are using green tea polyphenols. When one goes back to these biomarkers and these strategies, one can envision that these biomarkers of aflatoxin exposure, be it the albumin adduct or the DNA adduct, could be important efficacy endpoints in primary prevention trials in order to demonstrate that exposure has in fact been reduced, as well as the trials that we've been involved with to explore the efficacy of a number of chemoprevention strategies in these high-risk populations. I would submit for a number of you in the audience who might be interested in this field that utilizing this particular biomarker, this longer-lived biomarker of aflatoxin-mediated damage, could be and should be applied as a prevention endpoint to explore through looking at longer-term prevention strategies in these high-risk populations. The vaccination against HBV virus is a compelling and important public health goal for which not only is this supported by virtually all the health organizations around the world, but it is woefully underfunded and needs the political will and the economic support in order to implement a strategy that has already been proving to work in Taiwan. So again, this is something that, again, we need to be compelled with to further those efforts, but at the same time, there are other antiviral interventions that are either available or should be considered as part of our armamentarium in those individuals who escape, either are not successful in being vaccinated or have the misfortune of missing that opportunity for vaccination. This particular mutation in codon in 1762-1764 is something that I feel is a mutation that may allow us to address and identify the exquisitely high-risk groups of individuals for both screening and intensive interventions. And in fact, it is perhaps these people and others in using these types of biomarker strategies that really can allow us to really marshal and target our prevention resources, knowing that in many of these high-risk populations, we need to target those individuals at greatest risk for disease, and I would submit that these individuals may be those people that we need to target in our prevention and screening activities. So I'm going to come to summarize where I think we are now. These are the real mortality data from Qidong. You can see that by 1 year, greater than 90% of the people following diagnosis of HCC have succumbed from the disease. These outcomes have not changed in the last several decades, and we still are at tremendous loss of having appropriate therapies and treatments for those individuals diagnosed with hepatocellular carcinoma, irrespective of whether they're in Asia or they're in Baltimore. I mean, this is still an extraordinarily high mortality cancer. These are the data that I shared with you with respect to why we, in looking at the epidemiology, was immediately struck by the need to partner and develop relationships with our colleagues in Qidong, because these are the individuals who are not only developing cancer at a much higher rate than other parts of the world, but developing cancer much earlier in life. And I would certainly submit that if we can just change that age of diagnosis from 45 to 70, as you find in Beijing, that would be an enormous public health accomplishment in this population. These are the data on the mortality trend from the liver cancer studies in Qidong. They've been following through the cancer registry mortality since 1961. You can see that probably through getting better ascertainment of the cases, there was an increase in HCC mortality up until around 1990. But essentially for the last 20 years, HCC mortality rates have remained flat in this high-risk population. So let's project to the future. If we were able to get the types of intervention using the HBV vaccine that's already been successful in Taiwan and apply that type of penetrance into the population, and then we ask some questions and do some simple calculations, over the next 50 years in Qidong, we would see about a 50% reduction in hepatocellular carcinoma. So 50 years of follow-up, 50% reduction. I would submit, on the basis of at least some of the data that I shared with you and others, that because of the role of aflatoxin, if we were to intervene with the strategies that we have available to us and blunt the aflatoxin exposure in this population, we could probably achieve that goal 10 to 12 years earlier. This would be an important accomplishment. This would be something that I think we all need to attain. But at the same time, for anyone here in the early part of your cancer prevention career, really wants to take on the challenge that this is what we could do with our available technology against vaccine. 50 years, 50% reduction, we should be able to do better. If we blunt aflatoxin exposure, we know that we can attain that goal earlier. And what I would submit as you work with your various colleagues and scientists and challenge them, let's see whether we can do and apply our basic science better to try and make these curves much steeper. So instead of having to wait to mid-century, maybe we can bring that down 30 or 40 years earlier. And this is the challenge in this high-risk population. So in summary, I would want to share with you that the development and application of validated, quantitative exposure biomarkers have really allowed us to tease out the major etiological factors in hepatocellular carcinoma. At the same time, the use of these exposure biomarkers in a quantitative experimental chemoprevention model has really established the efficacy for a spectrum of agents that we can apply in high-risk groups. And last, the validated biomarkers allow us to identify high-risk people for screening and prevention interventions in population at risk for this disease, and we feel that this is how we're going to be able to forward the public health initiative of partnering these biomarkers with the need to intervene in these populations. So in closing, we have a handsome lab, governed and guided and guarded by Chin. This is our guardian of the lab. He really exists, or it might be a she. The statue is there. And all the collaborators that we've been involved with over the years in this work are our colleagues at Johns Hopkins, our colleagues in the Qidong Cancer Institute, at MIT, the Shanghai Cancer Institute, at Dartmouth, IARC, and the University of Minnesota. This is a true collaborative, interactive effort of many investigators over the course of time. It has really been a lot of fun. We share the data, we talk science, and we most importantly ask, how can we transmit this into our populations that are at risk? And finally, without the financial support of the National Cancer Institute and the National Institute of Environmental Health Sciences, we never would have been able to do this work. And at the same time, I would also want to share, parenthetically, the wisdom of these funding agencies to recognize that cancer is a global problem, and those cancers that occur in Asia and Africa are every bit as important as anyone else's cancer around the world. So again, I want to thank you for listening to this sojourn, and thank you.
Are there any questions? Can we take questions?
Should know better than that. Sure.
Thanks. This is Carlo Maley from UCSF. I was wondering about the double mutant version of the hepatitis B virus. Is there any evidence that it causes larger clonal expansions in the hepatocytes? That is, does it increase the fitness of the host cells more than the wild-type virus does?
There are a number of experimental studies that have been done in hepatocyte cultures that have shown that this particular mutation in the protein that evolves from it leads to greater viral replication along with a number of other biologies, but that's only been done in a number of cell culture systems. So we don't really know a lot about what happens in the host in the liver at this point.
So it increases the fitness of the virus, but we don't know whether it increases the fitness of the host cell.
Correct.
Okay.
Thanks. More?
This is Jack Lee from MD Anderson Cancer Center. First of all, thank you for a beautiful talk. But there's one thing that you haven't touched, that is the susceptibility. So they are, of course, they are HBV prevalent and aflatoxin, and how about the susceptibility issues? That in terms of, not everybody exposed with HBV or aflatoxin develop cancer, right? So in terms of the genome kind of individual susceptibility, there's some data on that.
So I think there's a lot of toxicology that goes with aflatoxin exposure in people that cause a myriad of other effects as well, and not just liver cancer alone. I mean, this is an issue where to get to the point to actually be diagnosed with a liver tumor requires, one, living long enough, and two, to have all of the resultant biology all filtering down to this rather singular endpoint. And at this particular juncture, on the HBV mutation, mechanistically, we don't know how this is formed. So we can't really address at least part of your question about, what is there, or is there something really uniquely different about those individuals who form that and embellish it for the development of disease. In the case of aflatoxin, I think a lot of this right now really gets summarized in terms of what the exposure is. I mean, there are clearly polymorphisms that play a role that give slightly differences in terms of the amount of adduct that's formed and the like. But in general, the exposure overwhelms these effects.
I see. Okay. If I may, I'd like to congratulate again for this decades of elegant work. And if I may take a personal note, I really like to see that curve, going down quicker, as you mentioned in the last few slides. Actually, my father died of hepatocellular carcinoma at the age of 45 when I was 15. Okay, and I couldn't agree with you more that whether cancer happened in China or in Asia or in Africa or in America, it's all the same. And our job is really to understand what happened and take this knowledge to reduce the cancer burden to the world. Yeah. So thank you for your wonderful work.
No, thank you. That was well said. Thank you.